So it's my great pleasure today to introduce Professor Samantha Harris from the Department of Cellular and Molecular Medicine at the University of Arizona in Tucson. Dr. Harris did her Bachelor of Science in the University of Illinois before undertaking a Ph.D. in physiology and epithelial ion transport at the University of Michigan. She then went on to do postdoctoral research in the lab of Richard Moss, and that was really where she got into cardiac muscle physiology, after which she was awarded a scientist development grant, which, having been fortunate to receive one myself, was really a key moment in your career heading into academia. Became an assistant professor at the University of Washington before moving to University of California, Davis, where she was promoted to associate, and then went on to the University of Arizona, where she's been recently promoted to a full professor and well-deserving. She's been consistently recognized with funding throughout her career and currently holding R01s. In addition, and an AHA award. Importantly, she's a fantastic mentor, and many of her trainees are also funded through AHA, NSF, etc. She serves and has served on Biophysical Society Council, the International Society for Heart Research, the American Heart Association, and reviews for NHLBI study section, as well as NSF and the American Heart Association. The reason why I invited Dr. Harris here is because she really is, in terms of cardiovascular science, in my opinion, one of the most elegant cell biologists we have out there, and really takes on very difficult, challenging questions in really clever ways. A lot of us here are stuck on the membrane with our ion channels and gap junctions and think about that, and then afterwards the cell contracts. And Dr. Harris has really become the pioneer in the field of sarcomere biology with a focus on cardiac myosin binding protein C, and how mutations in this protein lead to diseases and sudden cardiac death. I just realized, embarrassing enough, I was complaining to her one-on-one about how difficult ribosomes are to work with, but the sarcomere is very difficult to work with, and Dr. Harris has implemented some really beautiful both biophysical and biochemical technology to swap bits of it in and out, to do actual force measurements, etc. And I think she's going to really turn some of what our concepts are on their head today, in terms of what comes first in terms of excitation, contraction, coupling. And without further ado, I don't want to babble too much. Please welcome Dr. Harris here.
Okay, thank you very much for that very kind introduction. So yeah, that sets up a lot of expectations, but we'll see where this goes. With that said, I'm super happy to be here to actually connect with people. You'll see from my talk that I think mainly about the sarcomere, and so we'll talk about what that is in a second. But yeah, I think that the exciting new project in my lab might lead to new connections, and I think it's highly appropriate that I'm talking to you guys right now. So just as foreshadowing it, most of this work is unpublished, just to let you know. We have a manuscript in revision right now. Hopefully we're going to resubmit it in the next week or two. But this is all kind of stuff that's been coming out of the lab that's pretty much brand new, although the project itself has been in the works for a couple of years. And you might see why when we get through it. So what I'll be talking about is cardiac myosin binding protein C and how it affects contraction. And so what that is, and I guess I'm a firm believer in putting kind of the punchline first. And so what I'm going to describe to you is this protein's role in affecting oscillations of contraction, and you'll see that in a little bit. But first off, myosin binding protein C is a regulatory protein of muscle sarcomeres. And so sarcomeres are the functional units of striated muscle for the med students and for graduate students. So striated muscles, of course, are your skeletal muscles, and the striations are the sarcomeres. So that pattern of kind of striped pattern represents the sarcomeres here. But your heart is also a striated muscle. And so I've been focusing primarily on myosin binding protein C in the heart, cardiac myosin binding protein C, but there's also isoforms in skeletal muscle that we're beginning to investigate more now. Okay, so myosin binding protein C, or C protein as it was first named and later renamed to cardiac myosin binding protein C, which is a mouthful, so I'll probably refer to it as C protein primarily through here, is a protein that's present in these muscle sarcomeres. And you can see it here as these longitudinal stripes that are located in each half of the sarcomere here. So the sarcomere itself, okay, so those are C protein. And the sarcomere itself, this is a cartoon of it, is bound by these Z discs, which have alpha-actin on them. And then this middle portion where you see the black here is actually the myosin, and it's myosin thick filament. And then you have actin thin filaments in between the thick filaments. And I'll have an illustration of this again in the next slide. But for the most part then, myosin binding protein C has been a protein that's been attracting quite a bit of attention these days for a number of reasons. So it is an affected, or it's a protein that's regulated in both health and disease. And so, for instance, probably you're most familiar with it when you feel that burst of adrenaline or that fight or flight response. That beta-adrenergic pathway of stimulation ends up in phosphorylation of myosin binding protein C, and it's partially responsible then for the increased force and rate of contraction of the heart. I meant to try to change this, and I didn't succeed, but mutations in myosin binding protein C are the most common cause of hypertrophic cardiomyopathy, so we'll talk about that a little bit as well. But hypertrophic cardiomyopathy is the most common cause of sudden cardiac death. So oftentimes when you hear of an athlete that's been succumbed to a sudden cardiac event on the athletic field, it's due to mutations in either sarcomeric proteins or myosin binding protein C. So we'll talk about that in a second. And then in addition to that, aberrant post-translational modifications, in particular phosphorylation has been really focused on, occur during heart failure. And myosin binding protein C is known to be proteolyzed under cardiac stress, so much that now people are investigating whether or not C protein in the blood could be a biomarker, could be a better biomarker for myocardial infarction, for instance, than the standard TNI, which is often used. The other thing that I want to mention at this point is that, okay, so this is kind of a proteolysis or a loss of myosin binding protein C. The mutations in myosin binding protein C that lead to HCM are most commonly truncation mutations, which end up resulting in a lack of protein, so a haploinsufficiency or a reduced amount of protein. And those two concepts will be important as we go on for the talk today. So this is just to show you that myosin binding protein C, along with myosin, are the two most common causes of hypertrophic cardiomyopathy. But overall, most of the other disease genes that are associated with HCM are associated with sarcomeres, these structures that I gave you a glimpse of before. So we're going to just have a slide or two then of a little bit more. What is a sarcomere? How do they work? So this is another depiction of the idea that we have myosin thick filaments and thin filaments, and that there's a sliding motion of the thin filaments past the thick filaments here. And that's propelled by, if we were to take a close-up of our cartoon here, a cycle of myosin interaction with actin at the expense, I guess I should do it this way, at the expense of ATP, so that myosin on the thick filament here reaches up and grabs the actin filament, and then it has the chemomechanical cycle where it actually pulls on the actin filament and results in this sliding motion or a development of force against that. So, but of course, your muscles aren't always contracting, so it's controlled at some point. And for skeletal muscle and for cardiac muscle, it's controlled primarily through regulatory proteins that are located on thin filament. So this is meant to show the thin filament, which is actin helix here, with combined with tropomyosin, which is this cord-like protein here, as well as troponin. And troponin C in particular binds to calcium, which is the activation signal for the muscle, and results in that movement of tropomyosin. So that tropomyosin moves and then exposes these myosin binding sites so that myosin can actually attach and undergo these cycles of interaction. turns out that even though this is pretty textbook, turns out that we're just finding out that the system is way more complex than even this, but I'm not going to go there for today's purpose. But it's just fascinating that a process that we thought we know so much about is turning out to still have more surprises. And that those surprises I'll hint at a little bit is that, that you can actually get activation of thick filaments too, and we'll touch on that just briefly. Anyway, that's the canonical sort of activation of this system. And just to put that in perspective, I hijacked Don Burz's slides, and where you'll see he usually has one sarcomere. I made a more realistic version in my mind of putting all the sarcomeres on a cardiomyocyte. But the idea here is that the calcium comes from the sarcoplasmic reticulum. So the activation calcium comes from the sarcoplasmic reticulum, such that there's an orderly series of events that we'll illustrate in a second, that you have an action potential that occurs at the membrane here that ends up in releasing calcium from the sarcoplasmic reticulum, which then activates the contraction that we know as a heartbeat. So again, an orderly series of events. And this is, Don kindly gave me his animations for this. So you get your action potential, calcium-induced calcium release from the sarcoplasmic reticulum, and that's the calcium that binds to the sarcomeres and generates contraction. And then relaxation is essentially a reversal of these events, and so calcium is pumped back into the SR or back across the cell membrane there. There he goes. Okay. So the point that I want to make from this, though, and this is where I'm trying to foreshadow for some results that I'll show you, and then I will speculate what they could mean. But the point to know right now is that cardiac contraction is a highly organized event in space and time. And so the action potential and those events that I was showing you actually proceed very methodically from one cell to another and from one region of the heart to another, such that all that coordinated activity and all that coordinated electrical activity results in the stereotyped EKG that we're all real familiar with in terms of cardiac function here. So, and this EKG is due to the coordinated action potentials of all these cardiac muscle cells. So the other idea that I want to kind of insert into this kind of beginning and thinking about myosin-binding protein C and these proteins are that hypertrophic cardiomyopathy is a disease of the sarcomere, but I also told you that it leads or is the most common cause of sudden cardiac death. So a big question in the field has been, well, how do you get changes in sarcomeric proteins leading to arrhythmias, which are typically associated with action potentials in that the movement of ions. So what's going on here with these contractile apparatus proteins that could lead back to having changes in action potentials? And that's kind of the $64,000 question. We still don't know. The party line or one explanation has been that there's remodeling that occurs in hypertrophic cardiomyopathy. So you get fibrosis or other changes in the heart structure itself that could lead to arrhythmia. And so that's one of the leading possibilities. But what I hope to do is at the end show you a new possibility that I'd like to begin investigating. Okay, so getting back to myosin-binding protein C and thinking about this protein, I took a cartoon from a colleague's lab down the hall, and then I inserted myosin-binding protein C into the picture here so that myosin-binding protein C I show interacting with both actin and myosin, and we'll come back to that idea in a little bit. But initially, giving you a little bit more information about myosin-binding protein C, it's a modular protein composed of 11 repeating domains labeled C0 or referred to as C0 through C10. And these are folded Ig or fibronectin-like domains, along with a unique domain called the myosin-binding protein C motif, are the M domains. So this wasn't really imaginative labeling for these. But what I will tell you is that myosin binding protein C was named for myosin, or its binding partner myosin, because it has not one but at least 2 sites now we recognize for myosin. And it was originally purified as, or originally discovered as a contaminant in myosin preparations. So it binds tightly to myosin. The C terminus of the molecule is really responsible for its co-purification with myosin, such that this end of the molecule binds to, and I'll show you this in a second, but it binds to this thick filament here portion of myosin, as well as it has another N-terminal region that binds to the S2 region of myosin, which is pretty much where these necks come, or where the myosin heads reach out from the thick filament. So this led early on to, so C protein was discovered in 1972 or 3, that area. And what came out very soon after this was a really nice idea of how this protein would work, is that by binding myosin in 2 places, and so I've got one cartoon shown here, where it binds to the thick filament as well as to this neck region, that you could imagine that myosin binding protein C could easily restrict the movement of myosin heads away from the thick filament and binding to actin, for instance. So that under conditions where you could phosphorylate this M domain, under conditions of, for instance, beta adrenergic regulation, that then you free those myosin heads that they can interact with actin more freely and speed contraction, the rate of contraction and cross-bridge interactions here. Okay, so, and it turns out, and this kind of dovetails with this idea that I was saying, that it turns out that the thick filament also has an activation that it goes under as well, that we're just really understanding in the last decade or so, that it turns out that there is an interacting head motif for the myosin heads that we didn't fully recognize in cardiac muscle and was first discovered in smooth muscle. So what this is the thick filament, or EM reconstruction of a thick filament with 2 myosin heads highlighted, 2 crowns of myosin heads highlighted. And the thing that's important here is that instead of having the heads drawn like I did, that they're kind of extending from the thick filament, what this is meant to show you is that the heads are actually kind of laid back down on the thick filament and folded in some way, and folded in a stereotype structure. So this has led to the idea that, and this actually corresponds with a brand new state of myosin activity called the super relaxed state that you guys might hear about, where myosin undergoes really, really slow ATPase rate cycling, very, very slow. So these heads are basically parked, and it's an energy conservation sort of thing. But it's led people to propose that because myosin binding protein C binds to this kind of neck region here, that myosin binding protein C could actually be inhibiting, or be stabilizing is a better way to put this, these interacting head motifs here, and keeping the heads on the thick filament and keeping them from interacting with actin. This actually has led to a kind of a unifying theory of how mutations in both myosin and myosin binding protein C could lead to HCM, because if you had less C protein, you'd get less stabilization of the myosin heads or greater ability for them to interact with actin. So that matches up with a lot of data showing both that people with HCM mutations in C protein have less C protein, as well as this idea that hypertrophic cardiomyopathy is often, but not always, associated with kind of a hypercontractile state. And similarly, then the idea is that mutations in myosin are along these interfaces that interact with either C protein or with the thick filament here, so that they are also inhibiting this interactive head motif. So this has been proposed by Jim Spudich at Stanford, and is now one of the things that, like, if you were to open up, or if you were to Google search or PubMed search myosin binding protein C, these are the sort of things that you'd come across. I'm going to tell you a slightly different story, though, today. And so that's based on the great tragedy of science is the slaying of a beautiful hypothesis by an ugly fact, which I found really ironic because this was spoken by Thomas Henry Huxley, who was grandfather of Andrew Huxley, of course, Nobel laureate for action potentials, as well as one of the first proponents of the sliding filament theory of muscle contraction. But while this was pretty much true for me for a while, the idea that this hypothesis that C protein is only modifying the relaxed state of the thick filament is not entirely wrong. It's not entirely slayed, but the whole picture is a lot more complicated than we initially thought. And so it turns out that when I was first starting out, as I said, this hypothesis has now been given a little bit more color in terms of we understand more about this interacting head motif. But the basic idea that C protein was interacting with a thick filament in an inhibitory way was around since this discovery of C protein itself. What I did when I got in my independent lab was I set out to test that hypothesis, and I thought, oh, I should have some negative controls. A negative control would be to throw actin binding in the mix and see, make sure that myosin binding protein C wasn't binding actin if I wanted to test its binding myosin. But what I found was that the N-terminal domains, and so this goes back quite some time now, the N-terminal domains of myosin binding protein C also bind to actin. So what's that about? And I won't go over all the data that we have obtained, but using multiple methods with multiple collaborators, we had shown over a period of time that yes, indeed, the N-terminus of myosin binding protein C binds to not only myosin, but also to actin. And so this led to considerable awkwardness in the field for a number of years, and I think we're working it out right now. But I have to point out that these observations that we made were not new. We looked at, and I'll show you what I mean by this, we looked at truncated versions of the protein that bound to actin, but for full-length myosin binding protein C, people had seen this, again, starting in the 70s, that myosin binding protein C would bind to actin. But those observations were maybe overlooked because they didn't fit into a nice picture of how this protein could work when you had this beautiful hypothesis of how it worked and everything was fine. Okay, so at some point, I think this was a diagram from 2009 that we published, we came up with a new working model of myosin binding protein C in the sarcomere. And remembering about the thin filament here, so if I'm saying that myosin binding protein C could bind to the thin filament, immediately had implications then that C protein could influence thin filament activation or relaxation. It could be a drag between the filaments, which had been observed before. Or something that we're exploring now is that myosin binding protein C could perform as a mechanosensor and actually communicate between the thick and thin filaments. And so right now, my pet hypothesis is that this mechanosensing function is critical for the thin and thick filaments to communicate their on-off states to each other. So we're trying to follow up on that, and I'll show you more about that in a minute. Okay, but for instance, just this idea that it could myosin binding protein C could affect the thin filament activation and relaxation is again thinking in terms of this model where tropomyosin moves off of the myosin binding sites here and exposes them in response to calcium. So we were hypothesizing that maybe C protein could have some effect in that scenario. So to test these ideas, and again, this is many years of research in my lab, we went after this regulatory domain of the protein, which seems to be really the heart of it. And we used truncated constructs for a variety of reasons within assays that range from just simple ATP assays in tubes to binding assays that I had already mentioned, and force assays. And I'm just going to show you one of these that we'll come back to throughout the talk then. And that's the effect of these N-terminal domains on force. And so the setup that we have is that we take these truncated proteins here, and we can add them to a portion of muscle, heart muscle, in this case a trabecula, that was permeabilized with a detergent. So the detergent permeabilization results in what's called a skinned myocyte. And the skinned myocyte means that all the membranes have been removed from this. So you get rid of the plasma lamella, you get rid of the sarcoplasmic reticulum and any organelles inside of this cell, or these multiple cells in this case here. So that what you're left with, and it doesn't come across, I guess if you got really close up here, you could see the striations for it. But what you're left with is functional sarcomeres without all the membrane components with it. But what that allows you to do then is to control the calcium, so you don't need to be activating the cell with an action potential. You can just subject the cell to a bath of calcium with the change in calcium and measure force produced from it. So what that looks like, for instance, if you look at the control curve here, and PCA is just like pH, so it's a negative log of the calcium concentration. So calcium is increasing, going to the right in this graph. And as you increase calcium, what you see under the control conditions is that you get this increase in force production. When you add this C1, C2 component, or this portion of myosin-binding protein C, you get a very different curve that's shifted to the left. And what's happened there is that you get this left shift that is so dramatic that if you consider an amount of calcium where you'd get no force under normal conditions, in the presence of this protein, you get nearly half as much of your maximal force. So it's a really dramatic effect that you get just simply by adding this protein. But I'll show you a couple of things related to that, but I want to plant the caveat right now, is that this is an in vitro study. C protein is present here, and so this C1, C2 isn't necessarily restricted to exactly where those stripes are that I showed you in the very beginning with the electron microscopy. So C protein is located in a highly specific area of each each thick filament, and so the C1, C2 here, this is an in vitro experiment, and we have no way of restricting it. So that's been a problem for not only my lab, but labs that study myosin-binding protein C here, as well as the stoichiometry and the localization probably aren't the same. So it's been a big question as to whether or not that leftward shift, or these activating effects of C protein as opposed to inhibitory effects, these activating effects actually occur in vivo. And I'll show you a slide about that in a minute. But before then, I just wanted to show you actually another collaboration that I have with investigators at Eastern Virginia Medical School in Norfolk. So I was just here a couple weeks ago. And what we're working on with them is to actually understand the basis for this dramatic activating effect of myosin-binding protein C. And going back to what we know about tropomyosin and its movement, the C1 domain of, this is cryo-EM from Vitold Galkin's lab, that this is a 2018 structure paper that I don't have, oh yeah, okay, that I do have cited here. But the idea here is that myosin-binding protein C, shown in purple here, or at least one of its domains shown here, actually is pulling tropomyosin away from the myosin binding sites. And so this has been a really productive collaboration that we just were awarded an MPI last year to continue and to really understand the basis of C protein's activating effects on the thin filament. So, and that's in collaboration with, this is Howard White, who actually does enzyme kinetics. And so this is that activating effect of the C1 domain, and then we can mutate residues and mutate residues that are predicted to interact with tropomyosin and remove that activating effect. This is just a slightly longer construct that basically shows the same thing. Okay, so, oh, yes.
The calcium concentrations that you showed in the initial,
Mm-hmm.
the activating concentration
I'll go back, but it's around 1 micromolar can do it. So, and it of course depends on the solutions that you're using, but the PCA 5-7 is about where most of them are.
Right, okay. So a PCA of 6 would be equal to how many micromolar calcium?
So that's no, it's not 1 micromolar, 0.1 micromolar.
And so the plots that you just showed us are PCA, below PCA, is that correct?
Oh, here with Howard's work? Yes. So these are, yes. Okay, so yeah, you can actually, and I'll show you that in another slide as well. So you can have high enough concentrations of these proteins and under the right salt conditions that you can get activation of force in the absence of calcium.
Okay.
Yes, and so the PCA for Howard's work is nominal absence of calcium.
Right, so it's kind of non-physiological.
This would be non-physiological, the conditions. On the other hand, that's part of the collaboration that we have. So Howard and Vitrold do the cryo-EM and the kinetics under conditions that might not be physiological, but ultimately I take things either into a mouse or into a sarcomere condition then. But that's an excellent point though. This is, it's really dramatic that you can get activation even in the absence of calcium. And I'll show you one more slide with that. Okay. Yeah, and please stop me at any time if you guys have questions. Okay, so the question, building up from the big caveats that I said that the truncated proteins, that their stoichiometry and localization weren't the same as what you get for a native protein in a sarcomere, it led us to want to know, well, are these activating effects meaningful? So again, it was a hypothesis that was not entirely embraced by the field, that C protein would interact with actin, and that it wasn't entirely embraced by the field that there were the activating effects that could be meaningful. So I won't tell you too much about this, but the idea is that we wanted to know whether or not these were relevant to heart muscle contraction. And so you can imagine a case where you could have this activating effect of the N-terminus of C protein that actually augments activation at the beginning of contraction. Or you can also imagine a scenario where you can maintain activation and slow relaxation at the end of a cardiac cycle. And the only way that you're going to find out what it does to a heart is to actually make a heart to study. So this is where animal models have come in. And we made actually 2 animal models to test this effect. And we used HCM mutations that we had previously found to affect actin binding. So these are the phosphorylation sites within the M domain, and downstream of the phosphorylation sites are 3 helices with a number of mutations that are associated with HCM in people. So these 3 helices are shown here with 2 mutations in particular I'll show you more of, L348P and E330K here. And what we went on to do was to study these mutations, and we found that E330K reduced actin binding affinity, so it increased KD, whereas L348P increased actin binding affinity. So we thought these would be great mutations to really kind of get at what effect that either an increase or decrease in actin binding would have on the heart muscle contraction. And are we seeing augmentation or are we seeing delayed relaxation? So, and this was the panel that I wanted to show you that under conditions that I showed you where we have a trabeculate at approximately physiological salt concentration, what you can get is that L348P in the absence of calcium, so this isn't shown here, but the PCA is buffered to PCA9 here, is that you can get this L348P activating contraction in the complete absence of calcium. So we wanted to know what happens then to mouse hearts with it. And this is a lot of work that I will just summarize with one figure. But basically we found, and this is normalized elastance, so this comes from pressure volume measurements over time, PV over time here. And what you see is that in this green band here that corresponds to the mutation that increases actin binding affinity, that you actually get a slight prolongation of the time of ejection. So that time that the heart is ejecting blood, or that contraction is maintained here. Whereas we were delighted to find that the E330K that has a reduced actin binding affinity actually reduced the time of ejection here. So we've interpreted this then that that these actin binding effects actually occur on kind of the tail end of contraction, possibly when calcium is declining. And the hypothesis that I'm following up on with that is that there's a shortening-induced deactivation of the thin filament that C protein counteracts. So this was really gratifying for us, and it really kind of points us to also all the, yeah?
Are these mutations then also implicated in diastolic heart failure?
So yeah, you know, because HCM has a big component, and that was actually exactly what I was about to say, is that a relaxation component of it is consistent with this, yeah. And E330K, actually in people, the decreased binding one is fairly benign. So a lot of people have it, and it's almost just kind of a variant that is a benign variant. So there has been very little disease associated with that, whereas the L348P seems to be more severe in the family it was found in.
Can I ask one more follow-up?
Mm-hmm.
You were looked at the Hill coefficient for calcium in contraction, that I would imagine that if you're actually, if I understand this right, if you're tugging on actin with this link, you actually get rid of the positive Hill coefficient of calcium inducing more unwinding for additional calcium binding to induce more unwinding, that you're actually reducing the Hill coefficient?
So I'll have to look. I haven't looked at this data because in a while, and yeah, I'll tell you why, but the Hill coefficient does change, but I don't know what direction it is. So yeah, okay. So, and I should speed up because I've been a little too chatty, but the new project that I want to tell you about then kind of deals with this issue that I'm not sure if you were looking at the dates of these things, but the last slide that I had where I showed you the effects of the actin binding affinities was like a 2013. And then by the time I had 2 mouse models and a move in between with labs and an additional postdoc, it was 5 years later, okay, before I could really test a hypothesis in the mouse. So I have been thinking about ways that I could reduce the time between going from in vitro to in vivo studies. And I came up with something that kind of fills a methodological gap. And so what that is, I'm showing you a sarcomere here with the position of myosin binding protein C. And I wanted something that could speed discovery. And so we came up with a cut and paste approach to removing C protein from sarcomeres. So the cut was fairly easy to think about. There is tobacco etch virus protease that is, its consensus site is very rare in mammalian proteins. So this seemed like a good protease if I wanted to treat a delicate structure like the sarcomere with something. Whereas with the paste, I came across this this was a 2012 paper where Mark Howarth's lab at Oxford had described some bacterial superglues, and what they were was he made a split protein pair of a domain from Saccharomyces, no, S. pyogenes. So I can't remember the actual full name of the organism, but he abbreviated it SPI for the complete domain here. And he broke the domain into a catcher protein as well as a SPI tag, a little peptide here. And the really cool thing about this is that basically when you bring SPI catcher and SPI tag together, they form a spontaneous covalent bond. So it's basically like magic. There is no enzyme needed for this, and you get a bond that's equivalent to the isopeptide bond that gets put out or is linked to ubiquitin to lysines, for instance. So it's a covalent bond here. So finding these two things, I could design then kind of the system that I wanted to do with my myosin-binding protein C in sarcomeres. So if this is those 10 domains that I showed you earlier with 8, 9, and 10 actually binding to the thick filament, what we did was we made gene-edited spicy mice, and I inserted a TEV protease site here, followed by a SPI tag sequence within them. So these mice, the spicy mice, have C protein, but with this 20 amino acid insert here that has a protease site and the SPI tag.
So how big are each of those, the spicy and the...
So the SPI catcher and the SPI tag, the holodomain is about the size of an IG domain. So it's not huge, but it's not tiny. The SPI tag is only 8 amino acids. The TEV protease site, I think, is 12 amino acids. Okay, so what that allows me to do then is in my TEV permeabilized myocyte, which faded out there should be, in mine it has a myocyte in the background. Then I can treat it with this TEV protease, because again, the membranes I removed, it's just a matter of adding TEV protease to the bath. And I can remove the first half of my protein here. And this again is the business end of the protein that we know of at the moment. And then I can add new recombinant domains here and recover a full-length myosin-binding protein C, albeit with the SPI catcher SPI tag inserted in this middle section of the protein. So essentially what this does is it allows me to speed up the process of discovery so that only minutes are required then to test practically limitless numbers of insertions, deletions, mutations, etc. So use the cut and paste approach in myosin-binding protein C in sarcomeres, and this is some of the real data from it. And so this is, again, the initial setup. This shows you actually the fluorescent localization of C protein in those two kind of parallel portions along the middle of on the ends of a thick filament of the sarcomere, but that middle area is the M domain here, or the, the M line for the sarcomere. These are the two Z lines indicated in red. What you see is that the homozygous cells look, for all intents and purposes, similar to the wild type. Then when I go ahead and permeabilize the myocytes and treat with TEV protease, what we see is a removal of this normal localization pattern, but it's unaffected in the wild type here. And the idea that this C0, C7 N-terminal portion of the protein actually is soluble and can be washed away, as shown here on a Western blot, because we can remove it just by rinsing the myocytes in a fresh solution. And then we can recover the localization of C protein by adding back the C0, C7 with the SPI catcher, shown here. But notice that control experiment where C0, C7 was added back without the SPI catcher, you don't recover those localized C protein bands. So it looks like it works. Oh yeah, and then even after washing, that stays, which is consistent with now it's been firmly affixed to the thick filament. Okay, so just a little bit just to show you what happens in terms of force that's produced. And so this is wild type. This is before the TEV treatment, after TEV treatment, and after addition of C0, C7 SPI catcher. And all these curves are superimposable on one another, and so that's expected for the control. What we find is for the recombinant protein that's added, or for homozygous myocytes that actually have the TEV protease in it, then what we get is a slight rightward shift. So that's shown here as this bar going down after TEV treatment, and then that's recovered when we add back C0, C7. What doesn't happen, though, is when it's phosphorylated, we don't get that recovery. And this is a very small effect. And so this is not the punchline of anything, but this is consistent with beta adrenergic effects to overall have a decrease in calcium sensitivity. And that's usually due to TNI phosphorylation. So when TNI gets phosphorylated, you see this sort of rightward response. And TNI is the protein that's usually accredited with this decrease in calcium sensitivity as part of the beta adrenergic response. So this is consistent with it, but not something that wasn't unexpected or expected. I just won't get too excited about it. What really got our attention, though, is force instabilities that we saw in the myocyte traces before and after TEV treatment. So the most obvious thing that you should see here is that there's mechanical maneuvers at a few places in this trace, but this is a trace of force during calcium activation. This is just one of those myocytes I've showed you in the beginning, now emerged in calcium, and this is some isometric force that it's developing. On the other hand, after we remove myosin-binding protein C with TEVP treatment, what you see for each of these mechanical perturbations is that the line of the force is no longer stable. It's no longer quiet, and the force is going up and down, and what you'll see in a minute is oscillating. So it's in this area throughout this, and this is, I don't know what the scale is. I think this is maybe 30 seconds of contraction here at plateau. But the bottom line is that after we add the recombinant C0, C7 back, we recover the nice force stability. So there's no longer any oscillations. So I've now mentioned it a couple of times that these are actually due to oscillations. And if you would look at this trace, you wouldn't necessarily see that. But now what I'm going to show you is an actual video of what's going on in the myocyte preparation. And to tell you what you're going to be seeing is that this is going to be a video that's playing. All three videos are going to play. This is the same myocyte before treatment, after TEV treatment, and after replacement with recombinant protein. And these will be 30 seconds of live video at a force plateau. And the video will just continue looping. So what happens then is that we've activated in constant PCA 5.8 in this case, whatever is near the half max for force generation. And again, I've got to find a way to show you that these are live videos. So there's 3 videos playing here. But what you can notice here is that the middle video, when the myocyte has been treated with TEVP, you get all these oscillations going. And these oscillations will continue for 45 minutes. We've got one calcium that usually activates contraction, versus here where we have the one calcium where you can get these cycles of oscillations. And that these are actually oscillations, excuse me, is just shown by, because you've got different areas in the cell that are oscillating at different regions. That's why your force measurements don't show oscillating effects. On the other hand, if you were just to look at the force vectors for the box regions, what you get is a genuine sort of sine wave for this, so that there's wave-like behavior of the myocytes. So spontaneous oscillatory contractions, known as SPOC, have been described now for quite some time. So they're a series of sarcomere lengthening followed by, or really short lengthening followed by slow shortening over time, shown here and gone over in a recent review. They were first described by Fabiato and Fabiato, that described them way back in the 70s. And Fabiato at that time determined that SPOC originates from the sarcomeres and occurs in the absence of sarcoplasmic reticulum, which we've also verified in MyPreps by using ryanodine, for instance. So these are oscillations that occur most frequently, I mentioned it, at near the midpoint for force generation, and that, again, calcium remains constant under these conditions. So for the Japanese group that have spent a number of years studying SPOC, They've decided that it occurs with multiple cycles of contraction and relaxation, as I've told you, and they've come up with a couple of ways that that could be generated. And I'm not going to really interpret our results in terms of their model at this point, but what I want you guys to kind of understand is that you can get something like this by having a balance between cross bridges that are beginning to contract and to shorten, and then to have a catastrophic sort of release, and then have a relaxation with that. And what balances that is something that we're actually pursuing in the lab here. But so that could be one underlying cross-bridge way that you get this. But what I really want to kind of present, and this is speculation, hypothetical, but I think it's really interesting, is that there's synchronous versus asynchronous oscillatory muscles that have been known. So for instance, there's flight muscles in insects that vary from 100 to 1,000 hertz. And if you consider the idea that I showed you at the very beginning, that one action potential leads to one calcium transient leads to one contraction, that's a synchronous muscle. And so what you see here is that you get one calcium, one impulse, or one action potential, one calcium transient, and one wing beat. But you're fundamentally limited in speed with that to about 100 hertz. So that's probably about the top of this system, in the sense that it takes time to move calcium in and out of that sarcoplasmic reticulum. So that was one reason why I showed you that animation in the beginning, is that it takes time to move all of these ions across the membranes. The other system that insects have come up with to have faster wing beats is an asynchronous system, where you get basically an impulse that allows calcium to arise over a threshold value, and you get multiple wing beats here. So just thinking about that, then, thinking about that as well as the situation that I just showed you with the Spock, that the idea is that maybe what's happening is that C protein is, the loss of C protein is converting the system into a more asynchronous sort of setting. So what's really interesting after that, though, is that phosphorylated C protein does not completely dampen these force oscillations. So again, this is the before treatment here. And again, notice that the force trace is very quiet, well-behaved muscle. Then what we see is that after TEV treatment, you begin to get these oscillations again. But the key thing here is force traces after phosphorylated C protein is added still maintains quite a bit of these oscillatory activities. So, and that's just shown here. And you'll see qualitatively, and this is where we really need to quantify this, and we're not there yet, but phosphorylation seems to dampen the oscillation, or having phosphorylated C protein there seems to dampen the oscillations, but not completely. So that's shown here. So this is just now comparing, this is the before, this is the TEVP treated, that you can see the oscillations are just kind of wicked and rapidly going across here. But then they persist when I add back C0, C7 that's phosphorylated. And again, in this case, this will go on for 45 minutes, an hour, these sort of contractions. So together, thinking about the idea that asynchronous versus synchronous is a thing that is a proven strategy, at least evolutionarily, and thinking in terms of, okay, if this is phosphorylation, and phosphorylation is part of the beta-adrenergic fight or flight cascade, how could this be actually helpful for a cardiac muscle cell, and what could this oscillatory behavior be doing in a cardiac muscle? So first thing that comes to mind is that it might be an additional mechanism for increased speed of synchronization and contraction of either multiple sarcomeres in a cell, or I don't have, for all the gap junction people here, I don't have multiple cells joined here, but it could be something that also could communicate across cells. But I'm interested in the hypothesis and moving forward with testing the idea that this is one way for the contractile apparatus to actually feed back and talk to the membrane and talk to the membrane transport proteins in terms of then, and we'll get to that in a second, but for instance, arrhythmias and what could go wrong with the sarcomere itself could be transmitted through oscillations in some way to mechanically signal changes in membrane potential. Okay, so in essence then, what I think that we were observing is that the loss of myosin-binding protein C uncouples, or its phosphorylation uncouples EC coupling. So that instead of the canonical one action potential, one calcium transient, one contraction, that we've converted to a system that you can have one action potential, one calcium transient, and multiple contractions with that. So to me, this seems to suggest potential for the thermogenesis. You can imagine that, as I showed you in the beginning, that the action potential and the cardiac contraction is organized in time and space. But if you have loss of C protein in disease or with aberrant phosphorylation that also occurs in disease, you can imagine a system where you could have a propensity to be uncoupled, which might have deleterious events under disease conditions. So kind of to sum up then, we've discovered this new oscillatory behavior that seems to be controlled by C protein phosphorylation. So under normal conditions, I'm proposing that these oscillations might actually be beneficial in some way for beta-adrenergic stimulation to increase speed or coordination, might provide some actually helpful, beneficial mechanical feedback too. I'd like to explore this. But then when phosphorylation is aberrant in, for instance, heart failure, this could begin to be deleterious. The dysfunction could also be prominent under proteolysis when C protein is cleaved under stress. And we actually don't have the experiments right now, but we've simulated in our new SPICY system or the cut and paste system, adding back just the proteolytic fragment, and it doesn't stop the SPOX. So it would seem to suggest that this is a possibility. And then under conditions of HCM, where patients have haploinsufficiency, this event could conceivably affect the action potential properties of those cells as well. What's really interesting is that new work has shown by Teresa Kraft has shown that there's cell-to-cell differences in the amount of C protein produced. So you can easily imagine where you could have a foci of cells or a few cells that don't have C protein or have low C protein next to cells that do, and that could lead to further dysfunction. Okay, just to sum up then, I hope I've shown you that, or talked to you about C protein being an essential regulator of actin and myosin interactions, and that whatever you read, know that it also interacts with actin as opposed to myosin. We came up with a new cut and paste method that shows that without myosin-binding protein C, there is oscillatory contractions in sarcomeres, and really highly interested in exploring C protein's role in this, and also understanding whether or not SPOC is regulated by other proteins. And that this phenomena, this oscillatory contraction, might be something that is a new phenomena within cardiomyocytes, as well as skeletal muscle cells, and I've got other ideas on that, that actually would have implications for both cardiac function under normal circumstances, and then, as I've shown you, in disease situations. So with that, I'd like to thank the people who actually did the work in my lab. So Nathan is a first-year graduate student, but he's worked with me since he was an undergrad, and he worked as a technician for a year in the lab, but he's done all of the data collection for force measurements. Hannah was an undergrad in the lab that explored a lot of mutations in myosin-binding protein C that I didn't show you. Kevin and Rachel are techs in the lab right now, and these are some recent past graduate students and postdocs. The people that did the work at EVMS, Vitold does cryo-EM, so not too far away from here. And Howard was actually a postdoc in the lab across the hall from Gerald Offer, who studied C protein, or discovered C protein in the 70s. So it's a small world indeed. So with that, I'll thank you and take any questions.
Very good. Importantly now, thanks to your introduction, Stephen, I don't need to teach next week. So that's good. Okay, Rob.
So I was fascinating about the spot behavior. I noticed it's about 1 hertz, and the mouse heart beats at 300, 400 hertz. So clearly there's not a one-to-one relationship between the kind of spontaneous activity that you see and the rate at which action potentials are generated. So I'm wondering how you're incorporating that into your thinking.
That's an excellent point. So I have to say that the temperature that we're at is 15 degrees Celsius, so we're low for a mouse heart. But there has been a correlation. The Japanese people have correlated the frequency of the Spock as it differs from mouse to some big animal, maybe cow or elephant or something like that. And there does seem to be a correlation between the frequency of the Spock phenomena and the heart rate. And so prior to us discovering that C protein is, affecting Spock, that was taken as some indication that Spock might be physiological, but up until now, there's been no data to suggest it one way or the other. I've taken the correlation with heart rate as to be related to the underlying actual myosin rates, so that myosin or ATPase is known to correlate with heart rate, for instance. That's the Reedy's work long time ago, or Barony's work long time ago. So to answer your question, I don't know that at the moment, but that will factor into it, and I think there's going to be a temperature dependence.
Yeah, I mean, could it be some kind of clock almost that helps reset
Yeah.
contractile proteins with respect to ion channels?
Yeah.
Does not necessarily match.
I like the clock idea. I like that a lot. Came across that for another set of proteins with the calcium, yeah. Other questions?
I'm curious with your force traces experiments and the control groups that are pre-treatments with the protease, are they treated with a detergent?
Yes, yeah.
So if you compare like just without detergent treatment, contractility or force and detergent treated, are there differences?
So yes, there's, and I can't remember which direction it goes. There are some differences, but basically you can't do the exact experiment because before the treatment with detergent, there's membranes there. So to activate it, and you have no idea how much calcium is in there, but there are differences between the skinned and the non-skinned or intact muscle. And I can't remember which direction it goes, but the skinned myocytes are something that are just a kind of standard for studying the sarcomeric proteins.
Have you looked at how the presence or absence, so if it's interacting with both actin and myosin, have you looked at how it's changing the biomechanical properties of the tissue?
Yes, I'd like to, that's very much where I'd like to go with this. So in particular, thinking about it as a drag and that there's a mechanosensor. So we're actually trying to put a mechanosensor or a force sensor in the middle of the protein and measure a difference in Fret signal, for instance, between the two filaments. But I also think that it could be affecting the communication for the on state of the thin filament and the on state of the thick filament. And I'm interested in that reaction. But that's probably going to be communicated through the C-terminus of the molecule, which at present I can't get at, naturally. That'll be the next project.
So the cut and paste technique is really cool. So I was thinking, so you have one end attaching to the actin and the other attaching to the myosin. So when you cut in the middle, are both ends still attached? It's just not thick anymore, or just one part falls off?
Okay, so we know that the N-terminus can come out. So we know it's not soluble and it doesn't stay attached to actin or myosin. So the part that I was a little kind of glossed over is that I showed you that myosin, or that the N-terminus binds to myosin S2, and then I kind of just cut to another slide and showed you the N-terminus binding to actin. It's still controversial in the field when each of those things happen. People are coming around to that, yes, it binds both S2 and actin, but the question is, well, it can't be binding both, or right now we don't think it binds both. So there's an implication that it's dynamic. And so actually part of the R01 renewal that I just got using this technique is that we're going to have FRET probes inserted into the N-terminus to really kind of dissect when and how it's binding to the actin filament and when it's binding to myosin S2. So your question is, when I cut, I don't know which side or where it's binding. All I know is that that N-terminus is soluble. I can wash it out. That either interaction isn't like super high affinity.
All right.
Okay. Thank you.